Hello, my name is Seungjun Ahn, and I am currently an assistant professor at Mount Sinai. So this talk is something about the differential network analyses, so DN analysis. So it is a method that comes from the network theory, comparing the topological properties, such as the degree of centrality or connectivity measures, between two or more graphs or the networks under two different biological conditions of individuals. So it could be those who are at the higher and lower risk of getting cancer, or it could be those who are with the asthma versus those who do not have the asthma. So motivation based on the recent review article, two existing methods that were newly developed to the DN analysis for analyzing the microbiome data. But these methods, they do not incorporate additional covariate information that may be associated with the host or the composition of the microbiome. Therefore, we propose our method called the Sophie DNA, and we named it after our family dog that passed away recently. So this is the algorithm figure that we pasted directly from our full manuscript. So suppose you have the M by P data set consisting of the OTU table and the metadata. Then the first step is we estimate the association matrix, which is the matrix version of the network through the sparse CC. And of note, we have also attempted the other measures of association, such as the CC lasso, spring, and species. But sparse CC performed the best in our method, and we have detailed about the other methods in our full manuscript. So please refer to that. And the second step is we obtain the network characteristics by taking the group-specific marginal sums of the association matrix of each taxa. Then we repeat similar, but now we use the leave one out sample, and this step 3 we call it a re-estimation process. And the step 4 is then we calculate the Jackknife pseudo-value using the what we obtained from the second and the third steps. And lastly, we then fit a robust regression regressing the Jackknife pseudo-value for each taxa and for each individual. And the p-values are calculated from the t-test for each predictor variable. And to control for the false discovery rate, we have utilized the q-values. And simulation part of our study. So we have two different simulation scenarios. One that we call multivariable setting. So in such case, taxa are impacted by the effect of both binary group indicator and the continuous covariate. Whereas in univariable setting, the effect of the binary group indicator only exists. And we have considered the following sample size and network sizes, ranging from 20 to 500, and the network size of 20 and 40 taxa. And we have also adjusted the effect size for the simulation study. And for that, we have used the sparse DOSA to our package. So under the multivariable setting, in our simulation, it shows that regardless of the adjustment of the sample size and the effect size, we see that our method Sophie DNA it maintains the same size of the precision and accuracy value. However, our method outperforms the other two existing methods in terms of the recall and F1 values, as well as when we also increase the network size up to the 40 taxa, we see the similar pattern that our method achieved the higher recall and F1 value, whereas while maintaining the similar precision and accuracy size comparing the other two methods. And in univariable setting, we also see the similar pattern as comparing with the multivariable setting. And here's a table that shows the computational cost / the time. So here, our method, SoapDNA, it took a bit longer compared with the NetComi. However, it shows that our method, as a result, took substantially shorter amount of time and time comparing with the MNIM. So after the simulation studies, we have also utilized and applied our method to the two different sample data sets. So one is using the American Gut Project data. So here, since it's a type of differential analysis, so we set that the main variable is the binary group variable, which was to indicate whether a subject had or migraine or not. And the additional covariates are as follows. And as a result, we found that the 6 taxa were significantly differentially connected between the migraine groups while adjusting for the additional covariate information. And at the family level, some of the DC taxa were associated with the active or severe C. diff infection and systematic inflammation. And it took about 12 hours on the high-performance supercomputer and with the reasonable computational size specification. And the second study, so the original study, the full data from the original study is available in the Microbiome R package. So here we wanted to demonstrate the capability of assessing the temporal change in connectivity measures between 2 time points, because in the original study, they have given the dietary change interventions on the study populations. So the measurements were taken before and after 2 weeks of this dietary change intervention. So what we have done is we estimate and re-estimate the association matrices for each time point and for each group. And we do that for each group, so that when we compute the network characteristics and calculation of the Jackknife pseudo-values, we simply take the differences between these association matrices for each group. So here, the main predictor variable is the binary geographic location. So whether a subject is from Philadelphia, USA, or from rural South Africa region. And additional covariates considered were the sex and the BMI groups. And from the analysis, 6 taxa were differentially connected between these geographic conditions while accounting for the additional covariates. And we found species that were associated with the prevention and management of inflammation, intestinal inflammation, and the fatigue severity scores of patients with the advanced cancer stage. And it took 11 minutes on the same supercomputer size specification. So conclusion, SoapDNA is a novel regression technique that adopts Jackknife pseudo-value for DN analysis in microbiome data, which can include more than one predictor and predicts features of connectivity of a network. We have demonstrated through Monte Carlo simulations that the SoapDNA consistently reaches higher recall and FM score value while maintaining about the same size of the precision and accuracy to existing methods, NetComi and MNIM. And the full manuscript is currently under review at BMC Bioinformatics, and it is also available as a preprint version. And here's the information of that preprint, which is available in the archive. And I'm also happy to share that we have also extended our methods work to the software work. So please check out our R package available in CRAN. And it's called Sophie, and they're all capital letters. And here the list of major references that we would like to share. And that's it. Thank you.